Okay, now we're on to the very fun part of translation. We talked about transcription, which went DNA to RNA. Whoops, not RNA, RNA. Okay, and the type of RNA we talked about was mRNA. Okay, that part is transcription. Now we need to take RNA into proteins. Okay, now this whole idea, this is called in science the central dogma. Okay, this was coined, I believe it was by Watson and Crick. The central dogma basically is central to this idea of DNA, and it's accepted by all scientists, the scientific community. Okay, so our learning targets, we're going to talk about how does information go from DNA to RNA and then protein, and how in the world does that result to a trait. Then we're going to talk about the process of translation, including the roles played by the different types of RNA. So, different types of RNA. Please note, don't write, you already have this information. This is just review. Okay, different types of RNA. We have mRNA, that's carrying the genetic code or the genetic information from DNA out to the ribosomes. We have the rRNA, that's what's making the ribosomes, and it's where proteins are going to be put together. And tRNA, which brings in amino acids to the ribosome. So we have already talked about mRNA. In translation, we're going to focus on rRNA and tRNA. Alright, so translation, we're going to start with mRNA and the genetic code. As scientists, we're taking a look, once finally they're accepting that DNA is the genetic code. It's what's responsible for, coding for all of our traits, the blueprint, if you will, of you and I. They first ran into this first problem. Okay, there's only 4 nitrogen bases. How in the world is that going to code for all the amino acids we know that we have? We know that there are 20 different amino acids, so if each nitrogen base was specific for amino acid, that would mean we'd only have 4 amino acids. If we did codes, or if we did just 2 amino acids, okay, then that would only give us a total of 6. It would only code for a possible 16 amino acids. Okay, so by realizing that a codon is 3, okay, 3 different amino acids. I don't know what was going on with my 3 there. We get the total 20 amino acids that we know of. Okay, so that 3 nitrogen bases is called a codon. It's a triplet code, so 1, 2, 3 codons, or 1, 2, 3 nitrogen bases makes what is called a codon. That's a word you're going to need to get very familiar with. Okay, there are 64 different mRNA codons, so there's 64 different combinations that we can have in mRNA of the different combinations between adenine, guanine, cytosine, and uracil. 61 code for different specific amino acids. However, there are a few that are redundant, or some amino acids that have different codons. Okay, so obviously there's 64 codons, and we're just talking about 20 amino acids. So some of these codons are going to code for the same amino acid. I'll show you a chart that'll make more sense in the next slide. This is important to have this kind of redundant code because it's going to help protect against mutation. So if one of the codons is mutated, then if you've got, kind of a backup plan or another codon that can give you that amino acid, then you're okay. Whereas if you only have one codon and that one codon gets messed up somehow, then you're screwed. Okay, there are stop codons, and I guess you can't figure out what stop codons mean. Termination, it tells it to stop. Okay, polypeptide, once again, polypeptide is when we have a whole bunch of amino acids bonded together, which is going to give us our protein. There are start codons, or there's one start codon, methionine. Okay, this is what's going to start translation. So you need to get used to seeing a chart like this or like this. The wheel is preferably, oh my gosh, take two. The wheel is my favorite, but I will teach you how to use for each one of them. So let's say I have a codon that is AUG. Okay, so in order to do this, I'm going to look first letter, okay, first base, A right here, second letter, so I know I've got to be somewhere in this, somewhere in this whole row. Second letter is U, so I know I've got to be here, right, because it's like finding coordinates on a map. Okay, and then my third base is G, so I find here, follow the row over, AUG is my start codon. Okay, if I did, let's see, UAC, okay, then I'm going to go U, it's got to be somewhere in this row here, okay, somewhere in this row over here. My second letter is A, so I'm looking somewhere in here, and my third letter is C, so my amino acid that I'm looking at is tyrosine. Okay, ignore this codon up here. Now, what I'd like to do is show you what I was talking about here. If you take a look at the chart, I have amino acid glycine, amino acid glycine, amino acid glycine, amino acid glycine. Okay, so glycine could be GGU, GGC, GGA, GGG. Okay, all of those are going to code for the same amino acid, glycine. Okay, so that's going back to what we were talking about, that redundant code, is that by having these multiple ways to code for the amino acid glycine, if one of these in an mRNA gets messed up, let's say that one, you still have 3 different ways that you can still get glycine. Okay, so here's the wheel. The wheel is personally my favorite because what you do is you're going to start at the center and move your way out. Let's see, let's start with C-A-U. Okay, so I'm going to start with C, I'm going to go A, and then from A, go to U, so CAU is going to be, I think it's histine, okay, and these here that you have, the very last outer part of the wheel, all of those are abbreviations for the amino acids. Okay. Now, we talk about tRNA. tRNA is this funky looking thing. I always thought it looked kind of like Rudolph with this big nose up there. That big nose, by the way, is an amino acid. Okay, tRNA is going to transport amino acids to ribosomes. It's transferring those amino acids from the tip of its nose, okay, to another amino acid that the ribosome then is going to bond together, creating our proteins. Yay! Okay, amino acids bind to one end up here, okay, bind to one end up here, and then the opposite end is what is called an anticodon. Anticodon is going to match up with a codon. That's why it's anticodon. The order of mRNA codons is going to determine the order in which the tRNA brings in amino acids. So, I guess before we get more in depth, we need to talk about ribosomes. Okay, so ribosomes are composed out of 2 units. You have a small subunit that is made up of 1 RNA and proteins. You have a large subunit. Okay, I am not so concerned that you know that it's 1 RNA and proteins and then 2 RNA, proteins, and enzymes. What I'm concerned about is that you understand that rRNA makes the ribosome. It's what makes up the ribosome. that the large subunit does have some enzymes in there, and that it's got your large subunit and your small subunit. Okay, so the large subunit is where we're going to have these binding sites. We have the P site and the A site. Hopefully I'll be able to show you guys a video that shows you transcription translation, and you'll be able to see a ribosome, and it actually will have 3 sites. We're just going to really focus on the P site and the A site, and I'll discuss the last one when we watch the video. Okay, so binding site for mRNA and for tRNA. So we're going to have the mRNA in here in between the large and the small subunit with all of their nitrogen bases here. Okay, and then the P site, this is where tRNA, carrying polypeptide, comes in here. The A site is going to bind the tRNA carrying the next amino acid. Okay, so there's the P site carrying the polypeptide in there, matches. Our next one the ribosome's gonna move to is A. Okay, so polyribosome, there are several ribosomes that are going to move along the same mRNA. Okay, it's not just kind of like how DNA replication did not occur at the beginning, go all the way to the end. A ribosomal unit can start, the small and large, can start any place that they've got their start codon, that methionine. Okay, so they're going to have multiple polypeptides being created at the same time, and as soon as that start codon is available, another protein's able to bind to it and start kind of copying right behind it, or translating right behind it. Okay, so 3 parts of translation. We have initiation, elongation, termination. Initiation, the small subunit attaches to mRNA at the start codon. The anticodon binds to the start codon. Remember, the anticodon is found on the tRNA. Then the large ribosomal subunit joins to the small subunit. Elongation, we're going to have amino acids added one at a time. Okay, the tRNA is going to break away after its amino acid has been bonded onto the next one. tRNA is going to break away, go find itself another amino acid. The ribosome is going to move forward 1 codon, so it's going to move forward 3 nitrogen bases at a time. Movement of the ribosomes is called translocation. Termination, this is going to occur at the stop codon. I showed you guys 3 different stop codons. When a ribosome hits that, the polypeptide is going to be cleaved, or is going to be cut by an enzyme from the last tRNA, and then the ribosome is going to break apart into its 2 subunits. Okay, so let's see. We've got a small ribosomal subunit is going to bind to mRNA, and tRNA, the anticodon here, is going to bind. We've got our amino acid. Then we've got the large subunit that comes in. We get in our next, or it's going to go in and it's going to start adding amino acids from the next tRNA molecule to come in until we start getting this chain, and where it's just going to be this cycle of one tRNA to the next, building this chain of amino acids that's going to become a protein. Note over here the stop codon. Okay, once the ribosome reaches the stop codon, it's going to be signaled to break apart, and then you have your brand new polypeptide. Okay, so that is how proteins get put together, because a polypeptide is a protein. Now, our next thing, if you take a look at this picture, it just kind of ties everything together. Okay, we start with transcription. Pretty much, I guess you're going to have to pause this because time's about done. You go transcription, you get your primary mRNA that all the introns are cut out, then that leaves the nucleus, ribosomes attach, which form this chain of amino acids, and I am out of time. Feel free to pause and read each of the steps if you need further information.